All right. All right, I think we're back in action. Now the question is, does this little pointer work? Ooh, that's fancy. Okay. Yes. 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 I would say primarily affecting kidney, antidiuretic hormone, going to be affecting blood volume as well as reabsorption of water in that collecting duct, that collecting tubule, by increasing aquaporins, right? Those aquaporins are those channels that help us hold on to or get rid of more water. That's your primary location that I would say. Could it affect other tissues? Slightly, but primarily it's going to be affecting that collecting duct of kidney. Yeah, yeah, absolutely. Great question. So intro to endocrinology. We're going to go into endocrinology today a little bit more in depth. The people that are online, I just did a chalkboard drawing that you could probably be happy you didn't have to witness, talking about the different hormones that are involved in our endocrine system. So first, though, we need to talk about processes of control. A lot of questions that are going to come up on your NCLEX are going to talk about how is the endocrine system controlled and what are some of these processes that happen on a cellular level. I'd say a good chunk of your questions on the endocrine system are going to be talking about those second messenger systems. So again, endocrine glands and hormones. Here we have a list of the different hormones that I put up on the screen or on the board in acronym form. So now you can see their definitions here for you. They will probably put them in either their full form or in their acronyms. So I would definitely know TRH, thyrotropin releasing hormone, CRH, corticotropin releasing hormone. You want to make sure that you're able to identify these from one another. And then you can see the hypothalamus releasing hormones and then the anterior pituitary releasing hormones. And so we have them all listed here. I don't have MSH on here. MSH is released from the anterior pituitary. It is typically seldom tested on your NCLEX. So it's good to know, but I have yet to see a question referring to MSH specifically on NCLEX exam, but it does come from your anterior pituitary. Posterior pituitary, your antidiuretic hormone, otherwise sometimes known as vasopressin, and oxytocin. Your thyroid hormones, you have your T3 and your T4, your thyroxine and triiodothiamine, and calcitonin. Your adrenal cortex is also an endocrine gland, and it's going to be your cortisol and aldosterone are your big producers from there. Your adrenal medulla, your norepinephrine and your epinephrine. So components that can be both nervous system as a part of the autonomic nervous system, as well as a part of the endocrine system. Yes. Yes. Do we know how to do that? That's my next question. Is it over here? Behind me. Ooh, okay. All right. Ooh, all right. Do you want me to turn off this one too? Okay. Wow, I didn't break anything. That's good. At my other university that I'm at, if I touch something tech, I'll probably break it. So they know to not let me do it. All right. So then we have our adrenal medulla. Like I said, norepinephrine and epi, they have roles as both endocrine neurotransmitters or endocrine hormones, as well as neurotransmitters. We call these neurohormones, norepinephrine and epinephrine. They can be used in both those different systems. Pancreas, again, another exocrine endocrine organ. Its endocrine function is insulin and glucagon. Exocrine function is releasing all those digestive processes, which we'll get to on Friday. Parathyroid, we have our parathyroid hormone, and we have our testes and our ovaries, testosterone and estrogen and progesterone. Whew. Endocrine system, it has a lot to it, but really, if you can remember organ, hormone, you're going to go far. Because then if it's a condition of a dysfunction of that hormone, you already know the organ system or the organ or the location that you're working with, right? So we'll talk some about these processes of control, and we'll go in depth on a few of these systems. Almost all hormones, specifically endocrine hormones in the body, are stored in these secretory vesicles until they're needed. So different than neurotransmitters or some of these things, they're just released directly into the bloodstream. Hormones are stored until they get a signal, a stimulus to that receptor, and then they're released. Almost all of our hormones in our body are polypeptides or proteins. They're synthesized on that rough end of the endoplasmic reticulum. Prohormones, hormones prior to being activated, that's their pre-active state, are going to be transferred then to the Golgi apparatus and then packaged into those vesicles where they wait until they reach their until they hear their stimuli. Then they'll be released. Increased cytosolic calcium, so calcium inside the cytosol, due to that depolarization of the plasma membrane, so connections to that action potential, can signal or stimulate exocytosis of those secretory vesicles. So we do have some connections here. Or most commonly, you'll have stimulation of a surface cell receptor, which is going to throw us down the secondary messenger system of CAMP, our CAMP second messenger system, our cyclic adenosine monophosphate, activation of protein kinases, and then the hormones released. We're going to look at that a little bit deeper. There will probably be questions about second messenger systems on your test. So that's where we do have some cellular components that will come up. Mainly because it's just such a good, easy question because it's in so many systems, right? But first, let's talk a little bit about creation of other hormones. So steroid hormones come from cholesterol. So any of our steroid-based hormones are going to be, cholesterol is going to be your big building block. They typically aren't stored. They're lipid soluble. Your amine hormones, hormones that have that nitrogen in them, they're derived typically from tyrosine. Those are mainly your thyroid and your adrenal medullary hormones. Thyroglobulin is then going to incorporate thyroid hormones into that thyroid gland tissue itself. And thyroxine binding globulin is going to bind in the plasma. We're going to go into thyroid here today. So if these things aren't making sense, if you're not remembering thyroid metabolism, we're going to get into it. Clearance, again, anytime I give you an equation, I don't think they're going to make you calculate things on the NPLEX, but they will want you to understand kind of the idea about clearance rates, or there was a couple questions in the past about how quickly could a hormone be cleared from the system, or how quickly after stimulation or a stimuli and hormone release would then you see an end product result. So there's two major factors that you want to remember that increase or decrease the concentration of a hormone in the blood. The rate of hormone secretion into the blood, so how fast is it getting there from that vesicle or from the cell, and then the rate of removal. Well, that makes sense, and that's your metabolic clearance rate. Your metabolic clearance rate is your rate of disappearance of hormone from plasma and your concentration of the hormone as a whole. Hormones are cleared from plasma in several different ways. They can be metabolically destroyed by tissues. They can bind with tissues and be uptaken into those cells. They can be excreted by the liver into bile or excreted by the kidneys into urine, so your emunctories, right? Especially our steroid hormones, our lipid-based hormones, those ones you're going to see more excreted through stool, bound via bile. Your non-steroid hormones, non-lipid hormones, those can probably go in urine through kidneys. By far, probably your most common clearance is going to be by binding to tissues and then being either utilized or destroyed or recycled, repurposed. So some hormone receptors and activation. How does this process get started? This is common to the majority of these endocrine processes, right? So if we can understand in general how these processes are started, how these hormones are activated, you can apply it to almost all of these situations versus memorizing every single metabolic process, right? One blanket process, and then we tweak it based off of the system. So typically our hormone receptors and activation, all of our hormone receptors are typically on the outside of the cell membrane. This is for our protein, peptide, and catecholamine hormones, so that norepinephrine and epi. For steroid hormones, they're in the cell cytoplasm, so that's a difference. Why do we think that's the case? They can cross the border, exactly, because they're lipid-based, they're fat soluble, so they can get through and they can actually get into the cell. They don't have to bind externally. Some still do, but they can get in. And then some in the cell nucleus, specifically thyroid hormones. So they're going all the way inside those cellular follicles, right, for production. Thyroid hormones, T3, T4. So downregulation of hormone receptors. So receptor proteins themselves are inactivated or destroyed during their function and can change from day to day or minute to minute. This means that it's a very quick to respond system, which is great. Downregulation of receptors can occur in any of these following scenarios. So by inactivation of some receptor molecules, maybe conformational change of the actual site itself. Inactivation of an intracellular protein process, so thinking about your GTP, your G proteins. CAMP, inactivation of those processes. So downstream. Temporary sequestration of the receptor inside the cell away from the site of action. So this could be due to a conformational change of an enzyme, or this could be due to a competitive blocker. So a medication or another hormone that could also bind and temporarily inactivate or move that receptor away so it can't bind with its target hormone. Destruction of the receptors literally by a lysozyme. They're internalized, they're digested, or just decreased production. So any one of these factors can downregulate your hormone receptors. And if your hormone receptors are downregulated, your hormones themselves' ability to bind and then become active is going to be downregulated. So thinking about that when we talk about processes of control or systems of control of the endocrine system, you can apply these same techniques and tools to across the board. This is really helpful in pathology, right? If there's a pathological condition where there's a molecular antigen that looks like a hormone, it can also competitively bind and inactivate a receptor, then you're going to see decreased uptick of that hormone use. So the hormone receptor complex as itself. So we're going to talk a little bit about, in general, what these complexes look like. Essentially, the formation of this complex is going to alter that function of the receptor, and it can imitate hormonal effects. So what are some complexes? We have our ion channel linked receptor, or G proteins. These are by far the two most common, I'd say, we see. So ion channel linked receptors, you can think of all your neurotransmitter substances combined with those receptors in the postsynaptic membrane. They're going to change the structure of the receptor, like an opening or closing channel for ions. And they're going to affect sodium, calcium, potassium entry into a cell. So neurotransmitter-based substances, so norepinephrine, bind to these receptors, change a channel, and are going to affect intracellular concentration of typically sodium, potassium, calcium, our main ions. G protein linked hormone receptors, by far the most common. These are going to activate receptors that are indirectly regulate the activity of the target protein. So essentially a cell, we're going to look at a picture here in a minute of this process, but hormone binds to a receptor. It's going to activate a G protein within the cell, which is then going to trigger that response, that secondary messenger response, typically using CAMP, cyclic AMP, and then different protein kinases that are specific to that hormone action. Okay. Not worth memorizing every single protein kinase. Just know that it's going to be specific to whatever that hormone action was when it bound. We then have our enzyme-linked hormone receptors. These are proteins that pass through the membrane only once. The hormone binding site is extracellular, and the catalytic or enzyme binding site is intracellular. So there's an extracellular and an intracellular action. Tyrosine kinase, which is our leptin receptor, is a great example of this one. And then we have our intracellular hormone receptors that activate typically genes or gene production or gene function, because we're inside the cell. You can think of your adrenal and gonadal steroid hormones, thyroid hormones, vitamin D from calcitonin, retinoid hormones, a lot of these bind with protein receptors within the cell because these hormones, like we mentioned, are lipid soluble and they can pass through that cell membrane. They don't need a channel or a pore. Typically, these hormones that are intracellularly are going to change some type of gene replication or gene transcription and translation. They're going to have an effect on something within the genetic code. So this cyclic AMP system, adenyl cyclase, CCAMP. All right. So this picture here, if you have it on your computer, it'll be much easier to see because I realize that this is like a tiny little thing. But just to give you some purpose, cell membranes out here, cytoplasms here, and then within the cell further and further. So I'm going to walk you through a little bit step by step of what's going on. So our hormone outside, I have a pointer on the screen, that might be easier. There we go. So this is talking about T3 and T4, okay? Cell membrane here. Our hormone's gonna bind to the receptor. It's gonna couple that receptor to the G protein. Oh, this is an example of this isn't my CCamp even. This is my previous one. Here, let me go forward. There we go. This is my CCamp. So peptide hormone binds to a receptor. We have our extracellular fluid. It's going to activate a G protein typically stuck in the cell membrane. It's going to activate something like phospholipase C or CCamp. This is our IP3. Where is my CCamp? Oh, I'm so mad. All right, I have a picture on another slide. So we're going to have to, I want you to ignore this picture. This is not your CCamp mechanism. So I don't want you to get confused. I'm going to try to get me a little pointer. Okay, ignore this picture. I'm going to talk through the words, all right? Well, heart palpitation. I'm going to talk through the words, and then we'll see the picture on another slide. So I know I have a picture of a CCamp messenger system. But this is direct entry into a cell. This is not CCamp messenger. Okay. So what we're doing with our CCamp messenger system, though, but this is by far the most common system. So that's why I want you to really commit this one to your brain. Because if there's going to be a second messenger system used, it's probably going to be this one. Not always, but probably. So hormone binds to a receptor externally. It's then that receptor is typically coupled, meaning bound to a G protein, which is in the cell, within the cellular membrane in the cytoplasm. G protein then will become activated if it's the right thing that was bound externally. It's going to stimulate this process. It's going to use some energy, typically GTP to GDP. Then going to stimulate adenylyl cyclase, which is a bound enzyme within the membrane. That's going to convert a small amount of CAMP, cyclic AMP, inside of the cell. Cyclic AMP related to ATP, right? Adenosine monophosphate versus triphosphate. Related, different. CCamp then, or cyclic AMP, is going to activate our protein kinases, which is going to cause massive phosphorylation in the cell and activates all these little enzymes, which then equals a massive response. Essentially, small hormone amount, big cellular response, right? That's the beauty of the endocrine system. It's why it's such a good control system of the body. Phospholipase C, second messenger system, this would probably be your second most common second messenger system after cyclic AMP. The big difference with this one, it does have an external bound receptor. The hormone then activates a cascade of process inside, but it's going to specifically stimulate calcium ions that mobilize from your mitochondria, and your endoplasmic reticulum. So I'd say when in doubt, when thinking about the phospholipase C second messenger system, there's phospholipase C there. So think about calcium release internally within the cell that's going to stimulate that cascade of enzymes. Versus your cyclic AMP system is going to be G proteins, use of energy, protein kinases, phosphorylation that's going to stimulate that enzyme cascade. And lastly, this is direct entry and binding. That's what the thyroid hormone was. This is what the previous picture that was on the wrong slide showed. I don't know what's happening. But it's showing our T4 and our T3, our cell membrane. They get to come right on through. They did do some deiodinase. They removed iodine here. So we converted T4 to T3. That's then able to pass through into the nuclear membrane. We're going to go through this process kind of in depth here in a minute, but it's going to attach to the thyroid hormone receptor. It's going to have a direct effect on genes, right? We see gene transcription, mRNA, and then synthesis of new proteins that can affect so many things, right? The thyroid affects like every system. Just think about it affecting metabolism across the board, your thyroid. But the beauty of these hormones is they can act directly on genetic control. The bad thing is if they go wrong, they're acting directly on genetic control, right? Gene transcription translation. So when things go bad, they can go real bad. There's less feedback systems. Okay, that was the down dirty of intro to endocrine. Now we have the same language. That's the main purpose of this presentation. Now we're going to actually get into the nitty gritty. We will not go through all of this before I give you a break. Don't worry. I'm just going to start it. All right. So now let's actually dive into what these specific hormones do. Okay. We know some general terminology of the endocrine system. We know the organs and the players involved. We know some of the second messenger systems that they use. Now I want to delve into further what's happening at the hypothalamus and the pituitary gland level. So again, some ideas to remember. Our endocrine system is acting with our nervous system to coordinate and integrate activities within the body. These are your major response systems that help control everything within, right? If you were going to summate kind of in general what the endocrine system does, you could think about it as influencing metabolic activity via hormones. I mean, it does much more than that, but if you wanted to just try to put it in one little sentence in your brain. It has a slightly slower response time than the nervous system, but it's still fast. And it's going to control reproduction, growth and development, water, electrolyte and nutrient balance, cellular metabolism and energy, and our body defenses. So some differences between nervous system and endocrine system that I like to keep in mind. They will try to kind of confuse the two systems for you because they are both systems that respond and that help control the body in a myriad of different ways. So understanding how they differ will be important. The nervous system is a very fast response because neurotransmitters go directly into the blood, versus the endocrine system has to be activated by some type of second messenger activity, right? There's some type of binding and release that needs to occur before it can be directly stimulated. That being said, the endocrine system has a longer duration of response. So it's a little slower, but it lasts longer, versus nervous system is going to be quick to respond and short duration. Endocrine can act diffusely across the whole body, and they can act over long distances. Neurotransmitters just released amongst those synapses and really affecting that localized tissue. Endocrine hormones can be affecting the whole body everywhere. They're released up here and can affect you down here. We're talking mainly about endocrine glands. These produce hormones and they lack ducts, but exocrine glands, specifically think pancreas, right, has both, are going to produce non-hormonal substances like sweat, saliva, or digestive enzymes. And they have ducts to carry, not ducts like quack quack, but ducts with a T, to carry secretion to membrane surfaces. Love to mix up their exocrine and endocrine glands. So again, another summary slide to show you what the organs and glands are of the endocrine system. Hypothalamus, again, is a neuroendocrine organ, so it has both neuro and endocrine properties. The pancreas, the gonads, and the placenta all have exocrine and endocrine functions. They're both. And then there's a big collection of other tissues in the body that can produce hormones, but they're not necessarily considered as normal endocrine glands. And that's things like adipose tissue or thymus and cells and walls of small intestine, stomach, kidneys, and heart. So these would be non-endocrine glands that have endocrine function. So chemical messengers of the endocrine system, by far hormones are the most common that we talk about. Long distance chemical signals travel in blood or lymph. There's autocrines and paracrines though. Autocrines are chemicals that exert effects on the same cells that secrete them. So if cell A secreted hormone B and hormone B acted on cell A, that'd be an autocrine chemical. Paracrines are long-acting chemicals that affect cells other places. So if that same cell released chemical E and it acted on the cell all the way at your toes, that would be a paracrine. Autocrine, paracrine, they like to use those terms. The way that they might use that is talking about a cellular response or a neurotransmitter or a chemical release and saying that it's going to affect another system, some type of organ system. They might give you a list of like, is this an example of a hormone, endocrine hormone, an autocrine, a paracrine, or an exocrine secretion? They might list those all in one category and have you select and put it into a category yourself. Autocrines and paracrines, though, are chemical messengers. They are not hormones. So keep them separate in your brain. I know I misspoke and said hormone. But hormone's different than autocrine and paracrine chemicals. So two major classes of hormones, again, amino acid-based hormones. These are our amine derivatives having some type of nitrogen present. And steroids coming from cholesterol. These are our gonadal and our adrenal cortical hormones. There's this potential third class of eicosanoids, considered a hormone by some scientists, but some people more classify as a paracrine. Again, when in doubt, if it's not clear scientific information, it won't be on your NCLEX. So don't worry about memorizing that little nugget to brain. They're not going to include maybes on your test. 2 or 3 years old, right? So target cells are just tissues that have the receptor. Hormones are going to alter specifically the target cell activity. And there's lots of different actions that can occur. We've already talked about some of those. So main two separations, water-soluble versus lipid-soluble hormones. Again, different way of saying what we've already talked about, presenting the same information in a different way. Water-soluble hormones would include all amino acid-based hormones except for the thyroid hormone. They're going to act on receptors located externally on your plasma membrane, activate a G protein or some type of second messenger system, and cannot enter the cell directly, versus lipid hormones can. They can pass directly through, and they act on intracellular receptors that then are going to have a direct effect on gene function. This is the picture I was wanting to show you the first time. This is the pretty picture of CMP. So again, another example, hormone binds externally, it's going to interact with a receptor, it's going to activate a G protein, which is going to send some energy signal. It's going to convert either GTP to GMP or GDP or ATP to AMP. So some energy expenditure happens. Then going to activate adenylene cyclase, which is going to convert ATP to CAMP, which then is going to trigger protein kinases, again, specific for that hormone. And then phosphorylation occurs, essentially activation or chemical reactions that are activated by specific enzymes. Each hormone here has its own process that occurs at its target cells. They're not going to get down into the nitty-gritty of wanting you to know exactly what enzymes and exactly what protein kinases are used. They're going to want you to understand the processes of how they're different and similar. And again, review. We just talked about this. This is our calcium second messenger system and our intracellular system. So important, I'm saying it twice. Three different endocrine glands, not endocrine glad, although I'm glad that we're talking about it. Endocrine glands are stimulated to synthesize and release hormones in one of three different ways: humoral, neural, or hormonal stimuli. Humoral stimuli is by changing blood levels of certain ions and nutrients. They can directly stimulate a secretion of hormones. Okay? Neural stimuli, nerve fibers actually stimulating the release, especially important for our neurohormones. And hormonal stimuli, hormones that stimulate other endocrine organs to release their own hormones. So one hormone stimulates an endocrine organ to release another hormone. Those are your three major ways of stimuli through the endocrine system. So a little anatomy review. Our hypothalamus is in our brain. And it's connected to the pituitary gland via this stalk. The stalk is called an infundibulum. The pituitary, we've talked about the hormones it's secreted. We already have it on the board. But the pituitary has two major lobes, anterior and posterior. Anterior is glandular tissue. Posterior is neural tissue, which is why it contains neurohormones. This is a picture if you're visual, like me. You can see here hypothalamus up top, our infundibulum, and then our pituitary gland. You can also see that there's the paraventricular and supraoptic nucleus. So a little bit of neuroanatomy here. They're going to come down from our hypothalamus and synapse onto the posterior lobe of the pituitary directly. Again, encouraging the fact that posterior lobe of the pituitary is nervous tissue, it's neural tissue, versus anterior is a glandular tissue. Oxytocin and ADH are the two secreted hormones from the posterior lobe of the pituitary. What do these hormones do? We've talked about this already, but good summary, just to remind ourselves. Oxytocin, our strong stimulant of uterine contractions that's released during childbirth. Hormone trigger for milk rejection, positive feedback mechanism, acts as a neurotransmitter in the brain. It uses the PIP calcium second messenger system. This is one that binds and then uses that second messenger system with calcium release. Antidiuretic hormone. This is triggered specifically by osmoreceptors. They're monitoring for osmolality. If the concentration is too high, the posterior pituitary is triggered to release that ADH, which acts on the kidney and that collecting duct, increasing aquaporins, those channels, and helping us maintain an ideal concentration of salt and water in our body. It'll trigger essentially more water reabsorption. Release can also be triggered by pain, low blood pressure, and other drugs. So there is some pain, and specifically low blood volume or low blood pressure can trigger us to increase our blood volume by holding on to more water. This is where you could see this used in certain pathophys conditions and certain pathologies. High concentrations, though, can cause vasoconstriction, which is why it's called vasopressin. So if this process went wonky, if you had a syndrome of inappropriate antidiuretic secretion, you could potentially have a high blood pressure, or if you weren't secreting it appropriately, you could have low blood pressure. That's where you can see these effects occur. The anterior lobe is glandular tissue, which is great. That's what we're talking about with our endocrine system, is glands. It comes from an outpocketing of oral mucosa back in the day from embryology. So oral mucosa eventually is going to develop into this anterior lobe of the pituitary. It's vascularly connected to the hypothalamus, not connected via the same neurotransmitter processes, right? So posterior lobe has those direct nucleus that come down and snaps in that posterior lobe of the pituitary. Anterior lobe of the pituitary is not nervous tissue, it's endocrine, it's glandular tissue. So you're going to actually have blood flow that connects the two from the hypothalamus to the anterior pituitary. So those primary capillary plexus, hypophyseal portal veins, and secondary capillary plexus are your three major blood systems connecting your hypothalamus to your pituitary. Here's your hormones. Again, all but two are tropic hormones, tropins, that regulate the secretion of other hormones. So growth hormone and prolactin are really more acting directly on their tissues, and they don't have a lot of other secondary hormonal effects. The growth hormone is going to activate intrinsic growth factors within its target tissue, but it's not acting the same way as TSH or ACTH or FSH or LH does, where they activate further hormones down the system. All right, we're going to go through growth hormone, and then we'll take a break, okay? So growth hormone comes from our anterior pituitary. It's going to also be called somatotropin. You can see them used both ways on your test. Produced by your somatotrophic cells. It's going to have direct actions on your metabolism and indirect growth promoting actions. So it's going to be able to help with glucose sparing actions. So it's going to decrease your rate of cellular glucose uptake and metabolism. So think of it as the opposite of what insulin does. So it's going to keep glucose in the blood. So if you had low blood glucose, growth hormone can be helpful in maintaining adequate blood glucose levels from avoiding glucose uptake into the cell. It's not going to push glucose out of the cell into the blood, but it's going to keep glucose in the blood away from getting taken up into the cells. It's going to also trigger your liver to break down glycogen into glucose. So it's giving us more glucose from the liver, not normal cells, but specifically the liver. So that's good. We like that. It's going to increase our blood levels of fatty acids to use for fuel and encourage cellular protein synthesis, DNA, RNA transcription, translation, all of those processes. So I think of this as anything that can give us fuel to do more growth. That's what growth hormone's helping us do metabolically. Indirect actions on growth is going to trigger those insulin-like growth factors, specifically in liver, bone, and muscle, skeletal muscle. Those can then increase uptake of nutrients to give us more ability to synthesize DNA, then therefore proteins, et cetera, to allow us to do cell division. It also can help with formation of collagen and deposition of bone matrix to make our bones stronger. And it, in general, stimulates most cells to enlarge and divide. But really, if you're thinking about target tissues, your main two target tissues would be bone and skeletal muscle. It's going to target liver as well, but I would say if you had to focus it in more, bone and muscle, that's going to be your two major pockets for this growth hormone. All right. We're going to pause here, people online. We're going to take a 6-minute break just to have some time to stand because it's been an hour already. I know. Time flies when you're enjoying endocrine system. And then we'll come back and we'll finish going through the rest of these hormones and move into the thyroid. I'm going to stop recording people online for a moment, but then I'll come back to you.